This evaluation not just tells us, of course, which software may perform best, but also, that focusing on most abundant genera may be the safest option, and prioritizing sequences of markers that are more specific. We could sort of improve upon these methods a bit by really trying to increase the specificity. Also, for shotgun data, we're going to focus more on gene function analysis because, as was shown in a previous study published by Turnbow et al., is that certainly bacteriophyla can differ in different microbiomes quite significantly, but the functional categories will not change so much. And this is, I think, partly related to this problem that you can get a lot of species falsely identified as some other species just because of the fact that they're so sequence similar, but when you're actually looking at gene function categories, usually there is enough sequence divergence in functional categories to allow you to differentiate those better. So regardless, though, what's nice about this project is actually, while we care about that from a biological standpoint, being accurate in our analysis, from the standpoint of IDing markers, we actually don't even really care as long as we've got those markers, even if they are doing falsely identifying some reads. As long as we see that same pattern reproducibly, that can be good enough for identifying pollution sort of markers versus clean water. So just to show you some pilot data illustrating how that certainly seems to work. So here's, for example, some freshwater samples marked as S and marine water samples shown as N, where you can see the marine samples are very distinct from freshwater. As you'd expect, there's very different microbes in marine versus freshwater. But the S2 sample, which is fecally contaminated, clearly is very different from the S1 and S3 samples when they're clustered together. And in fact, it shows there is definitely a shift in the microbiome of the S2 sample versus S1 and S3 that involves bacteria, just looking at top 10 taxa, so looking at the most abundant species. We can really see a shift, and that gives us a lot of promise that we are really going to be able to find some good markers of these fecally contaminated water samples. Also, one point I'm really interested in is that certainly these taxa that are unique to S2 are potential new markers of fecal pollution, but also perhaps more interesting is that the absence of certain taxa may be just as informative, and there may actually be markers of clean water that we can use. And then finally, we can also compare these microbiome profiles to published fecal microbiomes from domestic farm animals and human and see that the S2 sample contain markers in common with the cow fecal microbiome. And so we can potentially find these species-specific markers of pollution source, which will really allow us to mediate these problems more effectively and identify the source. So in short, really what we're pretty hopeful we can do is through this bioinformatics analysis, careful bioinformatics analysis of this metagenomics data, factoring in some of these issues, such as I gave that example, we can select the most informative biomarkers and literally lead to development of a PCR-based test. The economic benefits are potentially significant. It's been shown how watershed protection saves money. In New York City, $4 billion in savings was shown to be possible for preventing epidemic illness. If you look at the impact of Walkerton outbreak, certainly outbreaks in general can have a huge drain on our economy. There's obviously environmental benefits in protecting aquatic environments and better targeting of post-pollution investigations. And then the social benefits. I personally really like take my kids to beaches, not have them get closed. So if I can keep some more beaches open, we can avoid 365-day boil water advisories in some areas due to high coliform counts that are actually not a sign of a pollution or not a problem. It's actually a risk. Certainly I would like to see that. And just generally, I think the concept, the philosophy of trying to be, like was brought up with the personalized medicine, trying to be a little bit more proactive, not just with how we treat human health, and trying to identify problems and risk factors early on. I think we want to do that also with environmental research too. So I just going to close with just bringing up, thanking Patrick Tang and Judith, who've done a phenomenal job project with Natalie and Jen and Bob Brennan, and there's this whole team of researchers involved in this project. And also I'd like to acknowledge people like Will, who just joined the BCCDC and were involved in some past human microbiome project work, and students in my lab, including Mike Peabody, whose work I predominantly mentioned, and also others that we're starting to form linkages with, and people like Richard Moore at the Genome Sciences Center, and also people like David Lynn and my student Amir, who are working on that pathway stuff. And I'll just thank you for your attention, and just mention that for funding sources, certainly Genome BC, Genome Canada, and really some excellent support from SFU to help enable this to go, and the Public Health Agency of Canada.